And now we welcome Professor Colin Campbell, who is going to tell us more about the detail level of the molecular understanding of the soil microbiota and how they are very much the engine room. They drive behind the processes and help the soils provide the very important functions of keeping our air clean and our water pure. Thank you, Colin, for coming along today. Chief Executive of the James Hutton Institute. Also had over 30 years of research experience within the area of soil microbiology as it's applied to agri-environment type systems. So can you tell us what it is about soil that's made you devote so much of your career to the subject?
Yeah, I think one of the things that attracted me about soil science was that it's a very multidisciplinary subject. You have to understand the chemistry, the physics and the biology. You have to understand the geography and the landscape perspective to really understand the topic. That was hugely interesting for somebody who's trained in one particular area to try and expand your horizons and understand all these other subject matters. So that was fascinating. But also it was its importance and the fact that not everybody could see its importance. So it felt really challenging to then say this is how important it is to everybody and demonstrate that to everybody. So it's been a fascinating opportunity to actually explore that and get that message across to people as well.
Thank you. Soil and the soil functions within the concept of understanding within the ecosystem services concept. It's interesting, isn't it? But can you tell us how much we currently know about soil biodiversity and the functions that relate to that and how it contributes to ecosystem services?
Yeah, I mean, we've always known a fair bit about how soil biodiversity contributes in terms of food production or in terms of supporting tree growth, for example, through symbionts and things like that. What we really never got to grips with was the extant diversity we find in soils and what so much of this mega diversity was actually doing for us. The advent of molecular biology techniques has really opened up our eyes about what they're doing in the soil. And far from being redundant in the soil, we're starting to find a whole slew of new mechanisms and roles for these organisms. And the functions that they perform for us in the soils are fundamentally important for the regulation of water, for the regulation of greenhouse gases, for the retention and modification of pathogens that move through the soil. There's a whole range of interactions which occur which actually give us functions and services that we never imagined before.
And things are moving at such a fast pace, and you are director also of the James Hutton Institute's postgraduate school. So how do you think that'll happen? What will be the next new thing in terms of our understanding in the next 20 years?
Yeah, I think we're already starting to see fascinating sort of outcomes of understanding the role of soil biodiversity in relation to plant growth, for example. Mechanisms whereby they actually communicate between different plants, mechanisms where they actually affect the flavour of crops. These are things we never imagined before, and I think we'll actually get even more understanding of how soil biodiversity influences every aspect of our crops and plants that we rely on for food security in the future.
And to help us understand that, what do you think will be the next great technological development in this field?
I think sequencing technology has always been going very, very fast. It's actually outstripped our ability to actually process the data. So much of the technological advances that we need going forward are actually to do with informatics and finding new informatic tools that allow us to visualise the data, understand that data, search it properly and find the kind of genes that we're actually looking for in this huge diversity of sequence information. So I think a lot of the technologies will be about information rather than necessarily about the sequencing side.
And how do you think we'll be able to handle that big data?
Again, the computing is growing to match that to some extent, but it may be actually need a bit of a revolution in the sense that we need to stop thinking about storing data. We always what we try and do is actually preserve the biology and the DNA so that we can always sequence it with new technologies. And this is why it's really important to have archives and germplasm collections going forward. All the information is there, and if you've got fantastic sequencing technology in the future, it's going to be easy to resequence it rather than actually store the data. So we could actually flip things around and actually not store data, but actually keep the data in the original organisms or the original sample and just continually always resequence it.
And do you think that will help us in the understanding and prevention of human diseases potentially?
Absolutely. I mean, soils are really important. They actually protect us. They protect the food chain from the entrance of pathogens. And soils are responsible for most of the antibiotics that we use in human health today. Even going back to the 1930s, the only soil scientists I know who won a Nobel Prize was Waxman, who found antibiotics in Streptomyces organisms, and yet here we are today, the same antibiotics are no longer of any use to us. We need to find new antibiotics. Soil is one of the places where we can find new antibiotics going forward in the future. And also soils, if we understand the persistence and survival of pathogens in soils, we can better protect human health going forward in the future.
So, well, the answer may lie in the soil then, Colin. But which do you think is your most favourite of these sort of multiple soil organisms that are there?
When you consider there are 10,000 species potentially in a gram of soil, it's very difficult to have a favourite, but everybody does have a favourite. It tends to be what, as a scientist, you've worked on the most. But actually one of my favourite organisms, the bacteria which are named Rhizobium. These Rhizobia bacteria are truly remarkable. They can fix atmospheric nitrogen and make it into fertiliser for plants to grow. They can live in symbiosis with plants in order to do that, but they're also extremely effective, what we call saprophytes, in the sense they can live in soil without the plants and still perform lots of functions. And for me, they're a very important organism in the soil.
Thank you very much, Colin.